这个我们今天呢是非常有幸邀请到了这个宾夕法尼亚大学的这个马乐园教授来跟我们分享他在这个增强 CAR-T 等方面的一些工作。同时呢，我们还有幸邀请到了许忠伟教授和张放博士呢来担任我们本次论坛的讨论嘉宾。其中呢，许教授呢他是非常早期参与 CAR-T 方面研究的，早在这个上世纪90年代就在日本从事这方面的工作。后来呢，加入这个卡尔顿实验室，并在这个第一例 CAR-T 治疗成功案例中做出了突出贡献。我想大家线上的各位可能都听说过那个白血病小女孩艾米丽她那个成功案例，许教授呢是背后的一个主要参与者。回国以后呢，许教授创立了细胞治疗公司，像这个叫贝塞尔特生物技术有限公司，落地在了北京，在一体 CAR-T 方面呢有诸多贡献。张放博士呢，他目前就职于药明康德汇源生物，是汇源生物的研发总监。他曾在这个北大医学院这个获得 MD， 在宾夕法尼亚大学的医学院呢获得 PhD， 有10多年的这个癌症及免疫学的相关研究经验。所以呢，我想两位行业大咖的加入呢，会让我们本次论坛的讨论呢可能会更深入、更精彩。同时呢，我们本次论坛呢还吸引了其他一些领域内的学者和行业专家。我们在此呢就不再做一一介绍了。下面由我对马博士的个人情况进行一个简单介绍，然后由许教授呢对他的主要贡献来做一个介绍。马乐园博士呢，他目前就职于宾夕法尼亚大学和费城儿童医院，是2家机构的助理教授。同时呢，他还隶属于多个 UPenn 和 CHOP 的研究所和研究中心，像比如宾大的这个免疫和免疫健康研究所，还有这个 Abrahamson Cancer Center， 还有这个细胞免疫中心，还有分子基因治疗中心。马教授呢是分别在这些研究所或者中心呢担任研究员。另外呢，据我所知，应该是马教授还在宾夕法尼亚大学的工程学院的生物医学工程系这个有他的一个职位一个 position， 他可以在那边招收这个生物学工程专业的博士研究生。在经费方面呢，马教授是拿到了 NIH New Innovator Award。这个是 NIH 对于年轻独立 PI 的一个很大的 award， 应该据我所知是一个1.5个 million dollar 的项目。同时呢，马教授呢还拿到了很多的私人的 foundation， 还有公益组织的 foundation， 像比如说这个 Schmitz Award， 这个曾经2017年的时候我也参与过，这个竞争非常激烈，这个 award。这个还有 melanoma 相关的这个 Young Investigator Award， 这个是也是竞争比较激烈。还有 Ben and Catherine Ivy Foundation Research Award， 这应该是一个跟 brain tumor 相关的一个 award， 都是实体流的。目前呢，这个马教授这边应该是研究经费比较充足，正在招收博士和博士后，所以呢，线上的同学呀，刚才这个免疫研究所那边，这个我们也发了一个转发了一个会议的通知，可能会有这个对于这些同学呢，可能是一个很不错的机会。另外马教授呢，他发表过很多的有影响力的论文呢，包括 Science 还有这个 Cell 这样的顶级刊物。他的主要贡献呢，下面主要由我们那个许教授来进行介绍。
好，谢谢这个宋教授的这个邀请啊，非常高兴能够参加今天这个论坛。马教授呢是我们这个宾大这个中国校友当中呢一个非常突出的一个新星，他这个是一个综合性的人才。我简单也看了一下他的研究范围，是个跨度也都挺大。这个重点呢目前集中在这个怎么样这个解决实体瘤的治疗，比如说 CAR-T 的应用啊。或者 microfiche 的应用啊等等这些领域。那么重点呢，就是说在肿瘤微环境这个方向，怎么样来找到一些好的突破口，找到一些好的这个 signal pathway，来进行一些免疫力调节啊，免疫这个敏感性的增强啊等等这些。尤其是今天他要给我们讲这个通过这个就是疫苗这个也是目前非常热的一个点，就 vaccine CAR-T 这些方面的一些新的一些 思路。我想呢，对肿瘤，尤其是实体瘤的免疫细胞治疗会有很多的启发和指引。那下面就欢迎马教授来给我们做精彩的报告。好，谢谢。
非常感谢徐老师还有宋老师，然后做的介绍。那下面的话我就开始做我的报告。Okay, so so I'm gonna like share the past work we have done in terms of using engineering solution to enhance the CAR T cell therapy. So especially for solid tumor. So before talking about CAR T and solid tumor, I want to briefly touch upon like different kind of immune cells and their role in cancer immunotherapy. So within a solid tumor, you will identify like many different type of immune cells, such as macrophage, natural killer cell, dendritic cell, CD4, CD8 T cells, and neutrophils. So among these different kind of immune cell, macrophage, neutrophil are mostly known for their pro-tumor effect, and natural killer cell and T cell for their like anti-tumor effect, and dendritic cell are mostly for their like propagating the anti-tumor immune responses. And any successful immunotherapy is often achieved by balancing the number and also function of these different type of immune cells, eventually leading to tumor destruction. So among all these immune cells, CD8 T cells are actually the major immune cell type are responsible for killing tumors. And this is mostly because they can directly recognize tumor and also release this effector molecule, such as granzyme, porphyrin, and interferon gamma, such as this different kind of like cytokines directly can acting onto the tumor cells to kill them. And this is one example showing the importance of CD8 T cells in the role of cancer immunotherapy. So typically in a very successful immunotherapy, if you deplete CD8 T cells, you will almost completely lose the therapeutic efficacy. This really shows the primary role of CD8 T cells in controlling tumor. So T cells recognize tumor through a very unique mechanism. So T cells have a T cell receptor. That's the major surface receptor enabling there to recognize tumor in a very specific manner. And the TCR typically recognize tumor by recognizing an antigen that's processed intracellularly in the tumor and then subsequently like presented on the surface of tumor cells in complex with another protein complex called a major histone complex complex. And this is a trimeric complex. But TCR will recognize like the peptide in this manner very specifically. And this initiates very specific T cell response. And because this very specific recognition and also super high sensitivity, it allows the T cell to recognize tumor even when the tumor only presenting 1 or kind of less than 10 this kind of peptide antigen on the tumor cell surface. And people have been taking advantage of this phenomenon by arming these non-tumor reactive T cells and giving them this tumor recognition potential by like genetically transducing such a tumor reactive TCR into the T cells. And now all the T cells suddenly become, tumor fighters. They can recognize tumor very efficiently and kill tumor very potently. So, however, there is there's some issue here. That's some of the practical barrier, which is like MHC restriction and MHC polymorphism. So as I mentioned just now, like T cell recognize antigen, not just recognizing antigen itself, but rather, when the antigen was present in a certain context, just like which in the MHC context. And however, within the population, within the human population, we typically have 6 MHC1 allele in each individual. And patient, although they may have like specific tumor specific antigen presented, it kind of expressing the tumor. However, this specific antigen needs to be presented by certain MHC. So this really limits the application of this type of therapy. So this actually leads to the CAR T cells, because the CAR T cells derived in a way that it kind of ignores this kind of MHC. Because the CAR was engineered by taking out the antigen recognition domain from antibody and then fuse it in a single chain format, which is single chain scFv, and fuse that into the signaling domain from the TCR complex, and eventually making it a orthogonal receptor, and making the T cell able to recognize target antigen in an MHC independent manner, and also serve as a leading therapy and have a breadth target recognition, just because the antibody can be engineered to recognize essentially anything on the tumor cell surface. So this makes the CAR T cell therapy really powerful and can be broadly applicable to essentially many different cancers. So this is just like showing some of the very early clinical data. like how the CAR T cell was able to eradicate tumor cells in vivo. So on the left you will see that's the first CD19 CAR T clinical trial data. Actually, the CD19 CAR T, once infused in vivo, they were able to rapidly eliminate the CD19 positive and CD20 positive acute lymphoblastic leukemic cells from the bone marrow, and eventually leading to more than 70% like patient with this tumor-free survival. And because of this success, like FDA have already approved like CAR T therapy, and now already approved 6 of different CAR T products. And among these 6 CAR T products, 4 of them are targeting B-cell leukemia lymphoma, and 2 of them targeting multiple myeloma. So they have numerous like other like CAR T clinical trials ongoing, targeting both leukemic cells and targeting both leukemia and also solid tumors. So however, for solid tumor, as you can see on the right, CAR T cells already been developed for solid tumor. However, there are many major issues associated with solid tumor therapy using CAR T cells. This is one of the examples. So this is also kind of one of the clinical trials initially carried out at the University of Pennsylvania. The patient bearing this glioblastoma, which having this EGFR variant 3 antigen expression. So this antigen is actually a very highly specific, like tumor-specific and highly specific tumor surface antigen. So which rarely can be found actually on solid tumor cell surface. So however, when the CAR T cell was infused into patient bearing this type of solid tumor, the patient still does not benefit much. And one of the reasons is actually the heterogeneous expression of this type of antigen on the tumor cell surface, eventually leading to the tumor escape, because like the outgrowth of the antigen negative tumor cells, which I'll mention, that's essentially the major goal of this, like the work I've done trying to address this issue to improve CAR T therapy against solid tumors. So, in order to talk about these hurdles of CAR T therapy in solid tumor, I want to briefly touch on like why CAR T has been very successful in leukemia and lymphoma. So other than, the antigen it recognizes, which is like B-cell surface antigen, there's also a couple other features. So first of all, the CAR T cell coexists with leukemic cell in circulation. And this makes like them to be very easily to recognize the target cell. And also, the regenerating B cell, healthy B cells in bone marrow can also stimulate CAR T cells. And this actually initial antigen stimulation of the CAR T cell really promotes CAR T expansion and also their early engraftment. And this engraftment persistence of CAR T cell eventually leading to the eradication of the leukemic cells and lymphoma cells from the patient. However, for solid tumor, as you can see, like most of the tumor antigen is really confined in this localized area. And this area actually have it's sort of very immunosuppressive microenvironment. And this limited antigen exposure and this immunosuppressive microenvironment eventually leads to limited CAR T expansion, persistence, and functionality, eventually results in limited efficacy of the CAR T therapy in solid tumors. There are additional hurdles, of course. So first of all, such as the lack of tumor-specific surface antigen. So many of the surface antigen you can find are tumor-associated antigen. They might be highly expressed in solid tumor, but they can also be expressed in healthy tissue. That makes like the CAR T therapy can have some on-target off-tumor toxicity. And another hurdle is that the antigen loss and tumor antigen heterogeneity. So this I already mentioned, like this instance in this glioblastoma. This is just like one of the very specific example I pulled out from a paper showing that the EGFR3 expression in glioblastoma can vary, between 23% to 100%. When the patient tumor really bearing 100% EGFR expression, they may benefit a lot from this type of CAR T therapy. However, when the patient bearing like on the lower end of the antigen expression, like 23%, their tumor would definitely relapse. So that's kind of the major work we're trying to address here. So talking about that, one of the earlier work we have done is trying to develop a vaccine to address like one of the major hurdle here, which is limited CAR T expansion, persistence, and functionality. And also when we were performing that work, we realized actually the vaccine boosting can even enhance like an antigen spreading event that it can potentially deal with this antigen loss of tumor antigen heterogeneity issue, and which I will also present kind of in this talk. Sorry, I think I got some notes here. Well, anyway, okay. So before talking about this specific CAR T vaccine, I'm going to talk about like how the conventional vaccination or how the conventional vaccine being designed. So and also the vaccine here we're talking about is mostly T cell based vaccine. So in the conventional vaccination setting, typically the vaccine formulation will include a protein antigen plus an effective adjuvant. The adjuvant is essentially like a danger signal that can activate the immune system to inform the immune system there's something danger coming on, and usually be activated to recognize it and eventually prime the immune system to fight against it. So typically the antigen will be a protein molecule, and when they formulate with adjuvant, both components will traffic through the lymphatic vessel to the draining lymph node. So once they arrive in the draining lymph node, and this protein antigen will be captured by the professional antigen presenting cell, which are typically dendritic cell. And once the dendritic cell captures this protein antigen, they will internalize the antigen and process them in the endosome into small pieces, into small peptide. And eventually the small peptide will be presented in complex with MHC and then be recognized by this T cell through this TCR peptide MHC interaction. And this interaction is actually served as signal 1, which defines the T cell response specificity. And then in the presence of adjuvant, the adjuvant will activate the dendritic cell, which will promote the upregulation of the signal 2, which is co-stimulation, which is essential for the optimal activation of the T cells. And then the adjuvant will also trigger this maturation and a secretion of different kind of inflammatory cytokine, which serve as third signal. So only in the presence of these three signals, the T cell will achieve optimal activation and differentiation, eventually like massive proliferation to deal with this incoming antigen. And I mentioned here, like this will be an MHC independent process because it's going through the natural like antigen presentation pathway, and the antigen has to form a complex with MHC in order to trigger T cell activation. But however, for CAR T cells recognize the antigen through a totally different way. Like it does not recognize antigen that is presented in complex with MHC, but rather it recognizes antigen that directly presented on the surface of the on the surface of the tumor cells. So this requires us to directly display the antigen on the APCs. But then how to achieve this? This is kind of, we use our synthetic polymer, which we call amphiphosphatase ligand. So this is also about biomaterial, but it's kind of essentially a lipid. A lipid like connected to a PEG linker, which is promotes solubilization. But then, we finally connecting to the ligand recognized by the CAR. So this is essentially a 3-component biomaterial. And because they having the lipid tail, once they formulate this material in aqueous solutions such as PBS, they're going to form a micelle. But the micelle is very unstable, especially when you inject it like in vivo. And this is because like there's a very abundant interstitial protein called serum albumin. This albumin have multiple lipid binding pockets. It will directly bind to the lipid tail of this polymer and then carry it into the draining lymph node. And once this lipid polymer got into the lymph node, it will come off the albumin and then jump into the surface of the APC. And then by directly inserting onto the lipid bilayer of the antigen presenting cell surface. And this way, it can be directly presented and be recognized by the CAR T cell, eventually trigger CAR T activation in the presence of co-stimulation and pro-inflammatory cytokine. This is essentially how this synthetic vaccine works to specifically trigger, and stimulate CAR T cells. So to eventually once we got this model design and concept kind of finalized, we want to test if it's true. So the way we did it is we took advantage of one of the available molecule we have, like which we call anti-phosphatase. That's essentially a lipid polymer bearing a fluorescent molecule, and then we did a tail-based injection. Once this molecule got injected, it trafficked immediately into the draining lymph node, as you can see here, which is the draining inguinal lymph node and the slightly distal axillary lymph node. They really lights up there. And when we isolate the cells from the draining lymph node, actually you can see all the molecules are labeled cell surface. So very, very efficiently. And then we kind of isolate other organs to see what's the other trafficking pattern of this molecule. Typically when injecting kind of different kind of chemicals or nanoparticles, they traffic directly into the spleen and liver. That's just naturally how the molecule traffics. However, as you can see, this vaccine molecule, it does not go there. Most of the signal is localized in the inguinal lymph node and external lymph node, really confine them in the lymphatic system. And also this surface presentation, as you can see from the confocal image on the bottom right corner, and this really shows this molecule can have very strong lymph node targeting potential and label the cell surface, essentially serve the purpose of directly label target cells and allow the CAR T to recognize and eventually trigger CAR T expansion. So then we kind of, once we know this molecule traffics the lymph node, we want to really dive in to understand where the molecule is actually localized within the lymph node. So we perform confocal imaging and also label, different lymph node structures, such as using CD3 antibody to label the T cell zone. Because in the lymph node, there are major compartments. One of them is T cell compartment, the other is B cell, and then the rest of them will be this called interfollicular region, which is mostly dendritic cell and macrophage kind of where they localize. As you can see, most of the signal actually really localized in the interfollicular region using this anti-50 antibody to really boost the signal of the anti-50 molecule. However, we really kind of perplexed by this phenomenon, but then we realized actually this kind of makes total sense, because in this, like the macrophage and dendritic cells, they're really phagocytic. They can capture a lot of molecules coming into the lymph node, and then directly, like because the lipid tail from the vaccine molecule, they can directly insert onto their cell surface. So this also got confirmed by the flow cytometry analysis, like on the right, which we basically like dissociate the entire lymph node into single cell suspension, and then analyze their physical display on their cell surface. And as you can see, most of the cell population that display surface physics are actually the professional APCs, including macrophage, medullary macrophage, and also the CD11C, CD11B double positive DCs. However, for T cell, B cell, and this lymph node stromal cells, they have a minimal kind of capture of the physics vaccine. So, now we know the trafficking pattern of this antiviral vaccine molecule. Now we want to know, does, this vaccine molecule can really, trigger CAR T cell activation? To model this, we took a model CAR, which we use like an anti-physic CAR. And then this anti-physic molecule, of course, now is served as target ligand for the CAR T cells. And by labeling the target cells with this anti-physic, we were able to see this like concentration-dependent kind of activation of T cell based on this interferon gamma ELISA, which is kind of an assay to monitor the secretion of interferon gamma molecule upon T cell activation. And because we see this like dose-dependent activation, this means this molecule really can trigger CAR T cell activation in a very specific and a very potent manner. So that was kind of in vitro data, but now we want to carry out some in vivo experiment to see if this vaccine can really serve as a vaccine, just like a natural vaccine, to stimulate CAR T activation and also expansion, of course. So the way we did this, we took advantage of this like congenic mouse model, CD45.1 and CD4.2. So we took the T cells from CD4.1 and transduced CAR into it, and then transfer them into CD4.2 recipient. So this way we can monitor continuously tracking the CD4.1 CAR T expansion in the CD4.2 mice by monitoring the CD4.1 marker. So what we did is, we injected like it's very low amount of CAR T cell, like only 50,000, and then give them like this anti-physic vaccine molecule or anti-physic plus adjuvant, which we call a complete vaccine. And as you can see here, compared to the control mice, which only received like 10 million CAR T, like when the mice receiving 50,000 CAR T then plus anti-physic molecule, you do see like significant expansion of CAR T cell. However, it's not as strong as the mice that directly receiving like 10 million CAR T. However, but when you receive this complete vaccine, when like with anti-physic plus this adjuvant, you see like massive expansion of CAR T cell at early stage, almost as comparable as directly injecting 10 million CAR T. And with second dose of vaccine, you can see even more potent CAR T expansion. And also this, like, once you're including this complete vaccine with adjuvant, you're not just including like massively increased CAR T number in the periphery, but also, significantly increase the CAR T memory formation, which is a central marker, so which is essentially like a very important marker to show that CAR T, the long-term persistence of CAR T cells. So why this adjuvant is kind of so important? So adjuvant essentially like can activate CAR T, can activate antigen presenting cell by stimulating antigen presenting cell to upregulate, this kind of many of the co-stimulatory ligands, which can serve as counter signal to trigger to promote T cell differentiation in a favorable way. And also this pro-inflammatory cytokine will also act on T cells to promote their proliferation. So in order to see, if our adjuvant, which is the cyclic GMP, which is a STING agonist, whether it can activate APC in the expected manner. So we tested like 5 different co-stimulator molecules on the APCs. And as you can see, all 5 different co-stimulator molecules got significantly upregulated within the first 3 days upon the vaccine kind of injection. So this really shows this adjuvant can really act on the APC in an expected manner to upregulate this diverse type of co-stimulator ligand. So even though the CAR T already have a built-in co-stimulation, but this additional co-stimulation can potentially help the CAR T expand and shifting their differentiation. So this is essentially the experiment we did to prove that concept. So what we did here is that we gave the mice this CAR T and then plus this complete vaccine. However, we've also give a group of mice with the co-stimulation blocking antibody cocktail. This cocktail includes like 5 different antibodies that can block the interaction of the CAR T cells with this different upregulated co-stimulator ligand on the APCs. So as you can see, once you administer this co-stimulator molecule, co-stimulation monoclonal antibody cocktail, you can block the CAR T expansion in the lymph node and also significantly reduce their functionality. So this really shows this additional co-stimulation triggered by adjuvant can play a very important role to achieve optimal CAR T expansion and differentiation. Okay, so now kind of we already tested this like synthetic vaccine in an independent experiment, but now we're going to come back to ask like how well this synthetic vaccine actually work compared to the natural vaccine. So in order to do that, we created this like bispecific T cells. We essentially took a TCR transgenic T cell. So this T cell is the clonal T cell population generated from derived from transgenic mice, and this T cell population expressed like this TCR, they're recognizing a GP100 antigen. It's called a PML T cells. And then we transduce the FISI CAR into it, essentially making it a FISI CAR and PML TCR bispecific T cell. So this way, we have the leverage that we can stimulate this population with either our synthetic vaccine to trigger T cell activation through FISI CAR, or giving them this traditional vaccine like GP100, trigger T cell activation through this PML TCR. So what you can see here is that, regardless of which vaccine we give, in this adoptive cell transfer setting, so both vaccine works equivalently well in terms of amplifying the CAR T cell expansion in the early stage, and also both vaccine were able to trigger CAR T memory formation. And this is exemplified by, when you stimulate T cells again using second dose or third dose of vaccine, which is separated by more than 30 days. So that's essentially enough time for the memory development. If you give them second dose, now you can see another spike, and giving a third dose, now you can see a third spike. So this really shows like, both vaccines were able to work like comparatively well. And this on the right, that's kind of actually the IVIS imaging showing that once we allow the T cell expressing luciferase transgene, just like show how much the expansion it can achieve with this vaccine. So just within 48 hours of after the first dose of vaccination, you can see almost the entire mouse kind of start glowing up. Basically, the T cells essentially expand such massively in such a massive scale, they traffic everywhere in the mouse body. Okay, so now we kind of showed in this kind of model CAR T and how well it works. But apparently, none of the tumor cell will express FISI molecule to allow the FISI CAR to recognize. Now we're gonna, to see if we can, use this concept to develop a bona fide to develop such a vaccine for a bona fide anti-tumor CAR. The way we did this is that we actually took a shortcut. We took like this like known, the antigen from EGFR3, which already have a very well defined kind of epitope, and which called a pathway3. And then there's no monoclonal antibody we use to generate this anti-EGFR3 CAR. And then, we typically follow our typical like biomaterial synthesis process, generate this like polymer vaccine, and then label the vaccine, so label target cell with the vaccine, and then stimulate the EGFR3 CAR T cells in vitro, and confirm that this vaccine can really potently stimulate CAR T cell very well in vitro. But now we're going to move to in vivo to see like how this vaccine actually works to stimulate this anti-tumor CAR T in the tumor-bearing mice. So essentially, the tumor we use here is called a CD2A. It is essentially a murine glioblastoma cells, and we basically implanted this tumor on the flank of mice, and then perform lymphodepletion, which is typical regimen we have to give the mice before adoptive transfer. And then, after CAR T injection, and we gave them single dose of vaccination, this amphiphile pathway3 vaccine. And then, we continue to monitor the CAR T expansion in the periphery. And what you can see here, so even though these mice have the antigen, which is solid tumor antigen, and just by injecting CAR T cell, you will see minimal expansion of the CAR T cell in the peripheral blood. However, in the presence of this vaccine stimulation, you can see massive expansion of CAR T. Just by single vaccine boosting, you can the effect will last till more than 3 weeks. And also, the vaccine, not just by increasing the cell number, but also changes CAR T functionality. So we perform like 3 different kind of assays to monitor CAR T functionality here. One is cytokine secretion, the other is kind of mitochondrial mass, the third one is granzyme B. So all these assays show that with vaccine boosting, you have increased cytokine secretion, increased mitochondrial mass, increased granzyme B level. This means the vaccine boosting not just give you more CAR T cell, but essentially give you better CAR T cells. So this is in the periphery, but then what happens within the tumor? So we also kind of create a different group of animals, and then we kind of to stop the experiment by a certain time point. That's essentially 7 days post vaccination. Then we take the take down those animals and extracted the T cells from the tumor and quantified how many CAR Ts are actually infiltrating the tumor. What you can see that like with the vaccine boosting, we see almost like 10-fold increase of CAR T infiltration into the solid tumor. And this infiltrated CAR T cells also exhibited like increased proliferation, which is kind of monitored by KRC7 staining, and as well as increased the polyfunctionality such as the granzyme B expression, interferon gamma TNF-alpha expression. So a lot of people have already shown like, the CAR T cells or other T cells, once they infiltrate tumor, they got exhausted or they got like dysfunctional. But you can see our vaccine can really kind of reinvigorate these anti-tumor T cells within the tumor. So we also perform like transcriptome analysis, essentially to understand like any major changes within the T cells, so why they become kind of resistant and still functional. So what you can see, the major signature we recover from this tumor infiltrating CAR T cell are actually this proliferation marker, like MYC target, E12 targets, but the rest of them are essentially all metabolic pathways. So this is super, super interesting. This means like potentially this metabolic like reprogramming by this vaccination really kind of make the T cell very potent killer within and can sustain their functionality within the tumor microenvironment. So now kind of come to like the final therapeutic study, and basically, like as you can see here, like once the tumor-bearing mice receive CAR T versus CAR T plus vaccine, only the CAR T plus vaccine group can really slow down tumor growth and eventually cure roughly 60% animals. And also, like, but till this moment, what we have saw is that with vaccine boosting, you increase CAR T number, also increase their functionality. But we also want to study, whether this qualitative change of the CAR T cell, this itself will be sufficient to control tumor growth. So that means we have to give, really control the CAR T cell number, but with one group going to have increased CAR T functionality. To achieve this, we essentially perform a secondary transplantation. And how we did this is that we first going to transfer CAR T cells into a donor mice that does not have tumor. Then we gave this donor mice either PBS or kind of the vaccine boosting. Then 7 days later, they extract the CAR T cells, then transfer equal number of them into a recipient mice that bears tumor. Now, this way we can monitor, because we really control the total number of CAR T cells we injected. This way we can really study whether this qualitative change of the CAR T cells can really define their anti-tumor efficacy. So this is exactly what we expected. So with the vaccine, by infusing this vaccine-boosted CAR T cells, we can really slow down tumor growth and also eventually leading to roughly 30% of cure. It's not as much as when you both giving more CAR T cells and making the CAR T cell more functional. But this is still kind of proved our point. Just like qualitative change of the CAR T cells, you will be able to control solid tumor more efficiently. The CAR T, any type of immunotherapy is different from the traditional therapy in a way that it will create immunological memory. So in the CAR T cell therapy setting, this immunological memory will essentially be CAR T cell memory. So that means if you re-challenge these cured animals with these antigen positive tumor cells, you will expect the tumor to be rejected. And this is exactly what we saw. So once we kind of challenge these cured animals with the EGFR positive tumor cells, all of the cured animals reject tumor, but none of the naive mice that never received the CAR T plus vaccine therapy. So then we ask the second question, whether we can challenge the mice with the antigen negative tumor cell. So what's going to happen? So this is what we observe. So the cured mice also rejects this antigen negative tumor cell. So apparently, because this antigen negative tumor cell, CAR T cell will not recognize them. So this must mean the endogenous immune system have been activated that are able to recognize additional antigen presented by the tumor cell that cannot be recognized by the CAR T cell. And this is actually through a process called antigen spreading. So during the antigen spreading, the initial CAR T action will kill tumor cells, will create tumor debris, and the tumor debris will be able to be captured by this migratory dendritic cell, which essentially transport and this antigen into the adrenal lymph node, and where they're going to be able to present the antigen to the endogenous T cells. And then once the endogenous T cell being activated and amplified, they're going to leave the lymph node and then go to the tumor site to recognize tumor through this natural TCR peptide antibody interaction, which is a sort of totally different mechanism than the CAR T. Now you have two different armies kind of attacking tumor. To verify this phenomenon, so we use interferon gamma ELISpot, which is essentially an assay you can monitor very specific and very sensitively to monitor T cell activation. The way we did this is that we treat them tumor-bearing mice with either CAR T or kind of CAR T plus vaccine therapy, and then we specifically extracted the host T cells by depleting the CAR T cell. And then they co-culture these like host T cells with tumor cells. The tumor cells, they will not bear the antigen recognized by the CAR. So this means every single spot you see here, that means it will be a host T cells recognizing tumor cells through peptide matrix interaction. And you can only see such a strong response in the CAR T plus vaccine treated group, but not in the CAR T or control T cell treated group. So this really means this vaccine boosting of the CAR T therapy can really engage the endogenous immune system to trigger this antigen spreading and potentially allow this process to more efficiently control the solid tumor growth and preventing tumor relapse. So once we know that, we essentially like trying to figure out what's the kind of major component here, because our vaccine composes like two different molecules. One is this amphiphile peptide-3 molecule, which is the lipid polymer. The other is this adjuvant. So we decided to really kind of disintegrate this process to understand like which component is important, or maybe both important. Both components have to be together to take this effect. And based on this result, essentially we kind of treated the tumor-bearing mice using either vaccine or CAR T plus amphiphile ligand or CAR T plus adjuvant or CAR T plus a complete vaccine. And what you can see here, really kind of only in the presence of complete vaccine, the CAR T therapy is able to trigger such a potent antigen spreading. So this really kind of signals to us that we really need this like complete vaccine to amplify this like very massive antigen spreading, and which can potentially use to deal with antigenetically heterogeneous tumors. And this essentially leads to our kind of next story. In terms of like how we can use this like vaccine boosting to control antigenetically heterogeneous tumor. But before talking about that, I want to mention like, once we found this, we want to study like how important actually this endogenous T cell are essential for control long-term tumor. So basically for the long-term tumor control. So what we did here is that we performed the therapy in two different types of mice. So one is wild type, which is the typical mice we're going to use. And now you can see, this mice, because this wild type mice, they still have their natural immune system. The CAR T vaccine therapy, you are able to control like most of the tumor. You're able to cure kind of 80% of the mice. However, the other type of mice are regular knockout mice. So this type of mice, they do not have an endogenous immune system. Therefore, there's no antigen spreading. And what you can see here, which is very interesting, that this CAR T plus vaccine therapy was able to control tumor growth at a very early stage. They were able to suppress the tumor to a measurable size, but eventually, all these tumors start to grow out. So, and when you analyze this kind of a relapse tumor, you realize actually most of relapse could be because of this antigen loss on the tumor cell surface. And this essentially explains like why this antigen spreading is important. Potentially, when the tumor burden is very low, they're able to deal with this like residual tumor cells very efficiently. Then, once we know this like endogenous T cells able to deal with like residual tumors, we also ask whether we can harness this phenomenon to directly allow the CAR T cells to treat a antigenetically heterogeneous tumor. The way how we model this is we use our EGFR3 negative and positive CD28 cells and mix them at defined ratio. And now you have a kind of range of like heterogeneity of the antigen expression. Then we implant this tumor into either wild type mice or regular knockout mice. And as you can see, like in regular knockout mice, kind of as long as they have like some portion of the antigen tumor, the CAR T plus vaccine therapy cannot control the tumor growth because they do not have antigen spreading. Therefore, do not have this additional endogenous T cell to help the CAR T to deal with the tumor cells. But in the wild type mice, so even like the 10% antigen-negative tumor cells can be efficient controlled. 6 of them can be efficient controlled, and even when the antigen-negative tumor reaches like 20%, it can still control 50%. And when it reaches like 50%, it can still control two mice. And even more surprising is that even when the antigen-negative tumor reaches like 75%, we can still control kind of 1 out of 10 mice. So this really shows us that this antigen spreading process can be harnessed to directly help the CAR T cells dealing with these tumors with pre-existing heterogeneity, which can potentially solve the issue that we saw before, like why this clinically EGFR3 CAR T therapy fails to cure solid tumor patients. But now with this vaccine boosting, we can potentially help those patients. So now kind of we know this like, CAR T plus vaccine therapy, they were able to trigger antigen spreading, but what's the underlying mechanism? So we have done like tons of study to figure this out, but this is essentially the summary of the key point here. So the way we did this is we blocked like the key effector molecule that's produced by the CAR T cells, and here just like cytokine. So we using this neutralization antibody to block TNF alpha or interferon gamma. So what we found is when you block interferon gamma, you'll almost completely abolish the antigen spreading. And to further verify this, we took we used like interferon gamma knockout CAR T and then perform the same kind of assay. And you also see like in the interferon gamma knockout CAR T treated mice, you also completely lost antigen spreading. So this all shows us that the CAR T derived interferon gamma is really the key or trigger of the antigen spread. So then we wanted to know like what's the kinetics. So when does the interferon gamma really play the major role? So the way we did this is we start using this neutralization antibody to dose the mice at different time points. And what we found is that actually the major effect took place at a very early stage of the CAR T plus therapy, maybe only the first week. And then you can see more potent suppression and spreading. But when you administer the antibody at the later stage, you do not see such a strong effect. And of course, when you're using this interferon gamma knockout CAR T therapy, you will see also loss of tumor control. So this really shows the interferon gamma is important for CAR T cells to control solid tumor, and also important for control to trigger antigen spreading. So because this antigen spreading process, we want to know using single RNA sequencing to see if we can really capture this phenomenon as well. The way we did this is using this sequel assay, which is one type of single RNA sequencing, but allows us to capture both transcriptome level and also the TCR repertoire level. So what you can see here is that after sequencing roughly 10,000 non-CAR TILs from the tumor, we were able to see with vaccine boosting, you significantly increase CD8 T cell population, but after administering an interferon gamma neutralization antibody, you kind of almost knock down the expansion of the CD8 T cells in the tumor. So once you do the TCR repertoire analysis, which is shown on the right, in the CD8 T cells, you will see there's a massive clonal expansion of the T cells within the tumor. So this really shows the sort of clonal expansion can be achieved within this antigen spreading process. But once you administer interferon gamma blockade, you almost completely abolish this clonal expansion. But for CD4 T cell, it's slightly different. For CD4 T cell, you do not see this very obvious clonal expansion, but rather based on the transcriptome analysis, you will see they have a significant phenotypic shift. So once the mice receiving CAR T alone, you will see most of the gene expression localized in this IL-17 pathway. This almost means the endogenous T cell exhibited a Th17 phenotype. However, when you administer a vaccine to boost the CAR T cells, now the phenotype shifts to more of a Th1 phenotype, it's present increase ITGB1, TCF7, CXCR3. That's almost like a signature for Th1 cells. And and also once you administer this interferon gamma blockade, you also kind of suppress the Th1 phenotype there. So this really means this vaccine boosting, not just induce clonal expansion and spreading to CD8 T cell compartment, but also kind of reprogram CD4, endogenous CD4 T cells to support the CD8 T cell response, to eventually leading to tumor eradication. Okay, so now we also perform a series of other animal studies using either genetic knockout animals or antibody blockade to figure out additional mechanisms. So here just a kind of overview of the summary of the result. So previously we already showed after blocking interferon gamma expression in the CAR T cells, we lost antigen spreading. But now we also know that in order to trigger antigen spreading, you have to engage dendritic cell, which is essentially the cell population to amplify the immune response. And the way we did this we either can knock out the key transcription factor called BATF3 from the mice, and then they do not have this DC cell, which is called presenting cell, which is essential for antigen spreading. Now you lost that. And then you will also kind of knock out this the interferon gamma signaling pathway from DC cells, or kind of specifically on the CD11C T cell, CD11C DCs, and you will see this significant reduction of the antigen spreading. So this means interferon gamma directly acts on DCs to trigger this process, and also the CAR T cell can also be a potential receiver of this interferon gamma stimulation. And once you knock out interferon gamma receptor from CAR T cell, you also lost antigen spreading. So this means interferon gamma can act on multiple cell population to sustain its effect in terms of triggering the antigen spreading. So DC cells also known to secrete IL-12, which can also act on CAR T cell to sustain CAR T activity. In the checkpoint checkpoint therapy situation. So we just want to test whether IL-12 is also play a major role here. So therefore, we using IL-12 neutralization antibody, which surprisingly just phenocopies what we saw from interferon gamma blockade. So this means IL-12 also plays a major part here, potentially sustaining CAR T activity and also sustaining this like positive loop here. So finally, because we previously showed the CAR T cell have a significant like metabolic reprogramming by vaccine boosting, and we also want to ask, whether this K pathway is essential for sustaining interferon gamma expression. So we basically knocked out one of the K transcription factor called the PCG1 alpha, which is essentially to maintain this oxygen phosphorylation. So what you can see here is that if you do knock out this PCG1 alpha, you lost interferon gamma secretion and also kind of mildly reduce or of this antigen spreading. So this means this oxygen phosphorylation plays some role, but may not be a predominant role here. Okay, so now we know kind of interferon gamma is kind of really the key molecule here to help the CAR T cell therapy to trigger antigen spreading. We want to ask whether we can genetically increase the interferon gamma expression within CAR T cells and to have much more like potent effect. So the way we did this is to transduce the CAR T cells with NFAT-driven interferon gamma cassette. This way, only when the CAR T being activated, and then they're going to have increased interferon gamma secretion. So this is what you can see from the bottom left corner, like this is the in vitro assay showing this CAR T cell bearing this NFAT interferon gamma cassette can secrete almost twice as much interferon gamma when they're being stimulated. And also when you inject in vivo these CAR T cells for this antigen spreading assay, you will see once just injecting the CAR T cell bearing interferon gamma, this NFAT interferon gamma cassette, you will be able to see increased antigen spreading. But once you add on another vaccine element, you will see even more potent antigen spreading. So this really shows like by just like solely increasing interferon gamma secretion, you will be able to have increased antigen spreading, potentially better tumor control. And then we eventually test this concept in a mixed tumor model. The tumor bears 20% antigen negative tumor that we that we previously identified as the optimal condition to model this. And as expected, once you give like the NFAT interferon gamma transduced CAR T cell and plus vaccine, you are able to control really kind of majority of tumors. Almost 80% tumor can get cured. So this is a brief summary of what I've mentioned so far. In the earlier work, we developed such a vaccine called amphiphile ligand vaccine, which can hitchhike albumin molecule, then be delivered into the adrenal lymph node, and eventually like being captured by APCs and trigger CAR T activation, just like mimicking natural vaccination process. And this vaccine boosting would able to trigger a very potent CAR T expansion, increase CAR T functionality. CAR T can attack tumor, producing tumor debris, and then eventually the tumor debris can be captured by dendritic cell, which can traffic the debris into the adrenal lymph node and eventually present this tumor new antigen to the endogenous T cell, which can essentially like initiate the antigen spreading process, allow this anti-tumor endogenous T cells traffic back to the tumor site to help CAR T cells to attack tumor together. And this is mostly through kind of CAR T derived interferon gamma and DC derived IL-12, a positive feedback loop to sustain this process. So here I'll have a few more slides, just quickly touch base on like in terms of how we can generalize this vaccine for any desired CAR T cell therapy. So typically the CAR T is produced by fusing the antigen recognition domain from antibody to the TCR signaling domain. And the antibody recognizes two different types of epitope. One is linear epitope, which we showed before using this EGFR1/3 CAR T model. But most of the antibody actually recognize the conformational epitope. So we took actually two different approaches. In the original science paper, we mentioned like both approaches, but here I'm probably going to only mention the second approach, which is we developed specifically for the FDA-approved CD19 CAR T. But basically, the CD19 CAR T is known to recognize a conformational epitope that is really hard to deliver using our vaccine platform. Then we hypothesized maybe we can produce a very short linear peptide, which is we call a mimetope, that mimics this natural epitope, but really have no resemblance to them, to the natural epitope in terms of sequence or kind of, but this interaction is essentially like just like biophysical chemical interaction that it can form. And once we develop this mime-tope, we can essentially conjugate onto our lipid polymer and allow the lipid polymer to deliver the mime-tope into the APC, then trigger CD19 CAR T activation and proliferation, and then allow CD19 CAR T to attack the leukemic cells more efficiently. So there's also a kind of major biological reason behind this, because when the CD19 positive leukemia lymphoma patient receive these CAR T cells, there's still kind of a bunch of patients experience this antigen positive relapse. So this is because the CAR T cell does not persist long enough. And actually, paradoxically, as you can see, this is one of the data produced, like presented earlier. They show that when the patient bear like less tumor burden, which is essentially less than 5% bone marrow blast, and the CD19 CAR T does not persist as much as those patients that bear more tumor burden. So this means this initial antigen stimulation is quite important to ensure the engraftment and long-term persistent CAR T, which might be essential for the long-term cure. So in order to develop this mime-tope vaccine, we took advantage of this Easter surface display. This is a high throughput screening platform that essentially allow you to display to present any protein of interest on the surface of the Easter cells. And then you can throw in a target that can potentially recognize these protein variants and eventually to isolate the protein variants that have a desired affinity. So how this process works is that you introduce random mutation to a protein of interest, and you can create essentially more than 100 million variants of the yeast. And then you can do this enrichment through a series like magnetic enrichment and effects-based sorting to identify the desired clones and then identify the protein variants. So for this mime-tope setting, we create a 10-mer kind of peptide, randomized every single position using this NK library design. And we got roughly 500 million variants. And then we use this FMC63 clone, which is essentially the same clone of the anti-CD19 antibody used for producing the FDA-approved CD19 CAR. And then we carried out this series enrichment and identified the mime-tope. And this is what we found. So we did find a very unique population from the library that once we identify the mime-tope exhibited a very high affinity, like 15.6 nanomolar. And this is essential because this is a very strong affinity able to trigger CAR T activation in vivo. And then, we first validated this chemically synthesized peptide, and then putting the peptide on top of the polymer and the label target cells, we show we are able to, trigger CD19 CAR T activation very specifically by monitoring the interferon gamma secretion. And then, we just created a hybrid model because to monitor this vaccine action in vivo, because we cannot directly use this vaccine test them on human CAR T cells in NSG mice, because NSG mice, they do not have lymphatic system. We cannot do vaccine study there. So therefore, in order to test the efficacy of this vaccine, we create a hybrid model by fusing this FMCC3 SCFV to the murine signaling domain. This way, we have a hybrid CAR. We can still do the functional study in the immunocompetent mice. So what we did here is that we label this hybrid CAR T cells with cell trace, which is a dye that you can monitor cell proliferation just by monitoring the dye dilution. And once you give them the vaccination and monitor the cell trace dilution, you can capture the cell proliferation. So with the control vaccine, you don't see much. You can still see very strong dye staining of the CAR T cells. That means they do not have much proliferation. But with so we also have another low affinity mime-tope vaccine, which triggers very minimal proliferation. But when we give them this high affinity mime-tope vaccine, we can trigger very potent cell trace dilution. That means the T cell proliferation. Okay, so this is all my work that I want to share with you. And this I want to give kind of acknowledgement to the people in my lab. We have a postdoc and 3 undergrad students, 2 bioengineering students, and then this is the funding resource. And most of the work has been completed at MIT, but also we also did some of the work for the revision for the upcoming Cell paper. So I think I also want to thank everyone who have been supporting me so far. And as Dr. Song mentioned, we have postdoc position available immediately. So whoever interested in this work, please reach out to me. Now I'm open to questions.
好，这个非常精彩的这个报告，马教授呢是第一时间将他们还已经接收，但是还没有刊出的工作给我们进行了一个呈现，这个非常的 impressive。所以我们看线上的各位专家、老师、同学有没有问题？徐教授，您有没有这个 想讨论的一些问题。
好啊，这个真的是在这个50分钟的这个讲演当中啊，这个信息量很大，这个我觉得工作做得非常好。然后就是说，在就是 CAR-T vaccine 这个方面可以说是开辟了一个新的一个方向，哎，用这种双亲的这种抗体，哎，来做这个。所以说这个我觉得非常非常好。那么就是说，我先抛砖引玉啊，就是有一些个别的问题先问了，然后看看大家有什么其他的问题啊，然后最后我们再进行一些讨论。那么就是说，我觉得你第一个方案呢，就是说，在这个通过这个白蛋白啊，这个这个lymph node的一个导向性啊，再结合一个其他特殊的一个抗原，然后给你 CAR 来验证呢，这个过程呢是可以 boost 这个CAR-T 的细胞的扩增，以及它的这个功能的，尤其在这个过程当中呢，这个 memory 含量还是相对比较高的，所以说可能导致这个 CAR-T 的这个 persistent 呀，还有甚至可能这个抗这个耗竭呀等等这些功能都是比较强的。那么这么就导致了你就整个的这个结果都非常好，赛德凯因分泌啊，这个包括这个K 的那个 function 啊等等这些方面都非常不错啊。那么就是说，在这个过程当中呢，就是说，实际上是激活了第三信号系统，对吧？T 细胞激活就是cytokine这一类的。那么这一类呢，就是说，除了就是说，这个 interferon gamma 这个你发现是比较突出的以外，有没有其他的这个cytokine的协同作用，或者是也很好？因为现在比如说在那 block 当中，你除了这个刺激以外，还有一些用那个这个PD-1 等等这种 block 的这个方案，哎，这个地方在这个方面你能不能比较一下？就是说你感觉哪一个更好？或者在特定肿瘤当中的更好？哎，这种情况能不能给大家讲一下，分享一下？
感谢徐老师的问题。其实这个机制方面的话，其实是就是说我们是测了很多，就是因为一开始我们不想把就是说这个就是这种 checkpoint inhibitor 这种 PD-1 抗体这种介入进来，就是因为我觉得他们的做效果是多元性的。但是所以说一开始我们没有做这方面，但是我们知道，因为 PD-1 抗体一旦介入进来的话，它是其实可以在淋巴结里面跟肿瘤里面同时更强的增活，就是激活这个 CAR-T。即便是我们的 vaccine 已经有效果了，但它进来的话，其实会有更强的效果，因为它是基本上去除了因为当 DC 被激活的时候，它其实也是会就是说增加这个 PDL1或者 PDL2的表面的表达。这样的话也会变相的给这个 CAR-T 一个就是抑制效果。如果说我们同时就是说给予这个 PD-1 抗体，那这样的话就是相当于是会给这个CAR-T 细胞一个更大的一个增强效果。但这样的话就是说，但是又会引入另外一个问题，就是说，因为那时候我们没法解释说我们这个 vaccine 到底是通过哪种方式来导致的这个效果。但是我想就是说根据像徐老师说的，以后的真正临床应用的话，其实我觉得还是再加入到这个 PD-1 抗体，还是会效果，我想一定会是更好的。至于其他的那些像一些细胞因子的话，我们检测了像TNF alpha，然后 interferon gamma，还有介素6、介素12，还有应该是一些其他的一些分子方面的。但像这样的话，就是说我们测出来是只有 interferon gamma 是效果是最明显的。但是就是说后面的话，就是我们发现就是因为就是 DC 它会产生介素12，这样的话我们就是在研究介素12的时候，我们会发现其实可以完全 phenocopy 这个，就是完全是模拟完全模拟interferon gamma 它那个 block 的这个效果，因为 interferon gamma 它是 CAR-T 本身产生的，而且我们也可以用就是说这个interferon gamma 缺失的这个 T 细胞来做 CAR-T，然后发现同样的效果。这样的话，基本上就是因为 interferon gamma 它是一旦 T 细胞被激活之后，它是基本上是第一个释放的因子，所以这样的话，就是我们的假设就是 interferon gamma 其实是一个诱导信号，就是一旦这个产生，那可能就是已经开始激活这个内源的系统，开始刺激这个就是这个 antigen spreading process， 或者是抗原分散这个过程。对。
非常好，谢谢。
那个张总，您有什么这个想讲的？
对，我觉得这个刚
嗯
才那个马教授的报告特别精彩，就是真的是从那个机制也研究的特别透彻，就是整个儿的，就是说比较非常的 frontier cutting edge 的这种思路和 technology，就是结合了 vaccine 来 boost CAR T。我觉得这个领域日新月异，也是这个做的非常的，就是说群雄逐鹿吧。然后我觉得就是我想的就是说这个。用这个很巧妙的这个 vaccine 的方法来去 synergize 这 CAR T，然后看到您说的两个 arm 这个 army，那么其实在这个两个 army 里面，到底就是哪个 army 更重要？所以我觉得好像这个 vaccine itself 就非常的 powerful 了，对吧？它可以去有一个 DC 的这种 positive feedback，然后它可以去这个招募和激活内源的 T 细胞。然后又能够 create memory cell，我感觉他自己很 powerful。然后我就说，如果说在您今后的这些 group 里面加一个 vaccine alone 呢，可能就更有说服力。就是说，它是不是 by itself 就可能治愈这个 cancer without even bothering CAR T？因为 CAR T 它的治疗还是非常 costly 的，也 risky，对吧？所以这个是一个问题。另外呢，就是说 CAR 本身呢也有一些这个，就是说可以说它的 efficacy。不是特别 easy 能够去被 optimize，因为最新的报道，这个王浩鹏和徐春怡教授对 tonic signaling 就是没有接触抗原 CAR T 能够自身 cluster 激活甚至 exhaust 的，所以需要一个非常 fine 的 tuning， 对吧？它的一个这个 antibody 这个 single chain FV 有一个最新的理论，从物理学角度看它的一个 positive charge density 这样一个指数，而且能够去优化它。能够非常好的控制一个最佳的 tonic signaling， 让它不会说没碰到抗原就能自动的去activate exhaust。所以在您的体系里，您配合了 vaccine 是怎么样去 fine tune control 或者说 manage 你的这个 CAR T 本身 tonic signaling， 通过 promoter 优化呢，还是通过这个 CAR 表达水平的优化来进行调控？
感谢张老师的这个问题呀。我先回答第一个问题，就是你刚才提到说能不能只用这个疫苗本身，其实是不可以的，因为我们之前因为疫苗本身它是应该说这个疫苗的话，我们只是把它这个本身的这个最小的这个抗原表位，就是 CAR T 识别的抗原表位放上去的。那这样说，这个本身的抗原表位它是不会激活内源系统的，因为它因为假定来说，我们把它放进，可能会被抗原 T 细胞识别，然后激活内源细胞。但其实这个表位它是 B 细胞表位，但是因为这个 B 细胞因为如果要生成就是说内源的抗体识别这个抗原表位的话，也是需要一个 CD4 T 细胞表位去作为一个辅助 T 细胞的。但是我们这个表位上是没有的，所以说你基本上只靠这个 vaccine 你是没法激活任何内源系统的。然后我们在第一篇的报道中，那个 Science 文章报道中，我们有提到这些，然后就是说我们也在就是说那个其中一个实验，我刚才就是我们当时第一次发现这个 antigen spreading 这个抗原扩散这种现象的时候，我们其实是检测过是不是能够把这个疫苗还有跟 CAR T 这个三方面，就这个两方面结合的事情，能够把它给打散，看哪一个成分起作用的作用。我们最后发现是，如果我们把疫苗中的那个佐剂拿出来跟 CAR T 结合，我们能够看到些许的，就是说那种促进这个抗原扩散。但是如果只拿这个疫苗分子的话，我们是没有任何效果的。然后 CAR T 本身也没有任何效果。然后你刚才提到是两个 army 对吧？就是说因为如果是要把肿瘤把实体瘤这么大的实体瘤给解决掉的话，其实你是需要一个很强大的一个 T 细胞群体来相当于是来铲除掉那个绝大部分的实体瘤细胞。然后因为这个抗原扩散过程它是相对来说需要一段时间的，那这样的话就是说在这个 CAR T 本身能够清除绝大部分实体瘤细胞的同时，这个缓慢来产生的这个内源的免疫反应也会去把那种剩残留的肿瘤细胞给清除掉，这样相当于一个互帮互助的过程。如果你只想依赖于这个内源 T 细胞，其实你是很难做到的，因为像现在很多的那种new antigen vaccine， 像那种叫做新生抗原这种疫苗嘛，然后像这种的话，这种肿瘤抗原疫苗的话，它其实是 是很有效。像我记得最近有一篇报道是BioNTech 用的 mRNA 疫苗做的那种肿瘤的抗原的疫苗，它是能看到效果的。但是就是说你需要首先你要找到这些抗原，那这样就意味着你可能需要花时间重新设计疫苗，而且你还得设计一个相对这种多元的疫苗。你如果只用单抗原疫苗，其实你也是没有这么好效果的，就类似于 CAR-T， 因为肿瘤可能也会丢失那个抗原。像第二个问题的话，你能稍微再重复一下第二个问题吗？因为我有点就是说忘记了。
对，就是那个 tonic signaling，就是说那个
对对对，
您是怎么去把控您的这个 CAR-T 有一个最佳的
嗯
tonic signaling，就是你有没有做一些这个它的一些 density charge 的优化？
像这个问题的话，其实我们没有特意放到这个 CAR 的这个设计方面，因为我们整个的过程就是完全是通过这个疫苗来调节已有的 CAR-T，就是我们所有用的 CAR，包括那个 EGFR V3 CAR，我们也是基本上是根据已经报道过的临床应用的 CAR，只是我们把它那个T细胞那个细胞内的那个signaling domain，我们那个换成了老鼠的。至于那个 SCFA 的话，其实也是当时临床上应用的那个，所以呢，我们就是基本上所有的完全是根据上临床来，基本上我们的目的就是设计一个疫苗就可以直接应用到临床上面。但是你这个问题其实问的挺好，就是说 CAR 本身如果是单纯用这种传统性的 CAR 设计的话，就是 SCFA 加上这个signal domain， 你可能会形成这个 cluster， 因为这个 SCFA 可能会它折叠的不好的时候，可能就会形成这个 cluster。但现在有新一代的，就是 CAR-T 设计出来，而且它不是像这种传统意义上设计，它是把这个是从 Sloan Kettering 出来的，它是把那个这个抗体的抗原识别区这个 VHVL 融合到那个 TCR 的抗原识别区，这样的话你就设计了一个相当于是它叫 HIT CAR， 就是 HIT。类似于就是把这个antibody 还有 TCR 一个 hybrid CAR， 这样的话就是你不会再有这种 tonic signaling， 因为它本身产生的话就是只是在一个单独的这个TCR complex 里面，但是现在它叫 hit complex， 这样的话，而且它可以识别抗原有更强的那种敏感度，相比于这种传统 CAR 来说，所以呢，就是根据像要回答你的问题的话，就基本上是我觉得像这种类似于 TCR 的 CAR 设计的话，可能是未来的发展趋势。这样的话，就是因为它首先有更强的这种灵敏度，而且又不会形成这种 tonic signal。
对，是的，
然后
对，非常感谢您的回答。我觉得是，这个就是相当于这两个 arm 实际上是一个 synergize 的关系，对吧？就是说它一定是要有 CAR-T 去在里面去这个把这个肿瘤细胞杀死 debris 啊，然后造成这个，我觉得这个是一个非常，就是一个很完美的这样的一个 system。刚才你讲的这个 tonic signaling 变成通过 TCR 也是觉得是一个 very make sense 的一个 rational design。然后我最后一个 question 就是说，就是在这个 loop 里面，您刚才讲到了它的一个多种 cytokine 第三信号，那实际上这个肿瘤细胞实体瘤很结实，你要把它彻底的一层打碎，这个穿透是很重要的。所以我说这里面实际 most likely involve 了 macrophage，就是说您的那个 cytokine 有没有看？刚才你讲的 IL6啊这种，是不是其实也 recruit 了更多 macrophage？因为它的这个对肿瘤实体瘤的浸润力是其他细胞的10倍，所以说是非常那个重要的一个这种对实体瘤的。所以说有 CAR macrophage 这些。所以您的这个体系里，我觉得除了你当时 T， 对你当时 macrophage 应该是也有 recruitment， 也有激活，对吧？
对的，你刚才这个问题很好，因为当时我们就是第二篇那个研究的话，那个 cell paper 其实有很多的那种这种肿瘤浸润免疫细胞的分析，包括 macrophage， 我们是也能看到macrophage 的变化，然后也有它表情的变化，但是就是说因为 T 细胞本身就是一旦被 vaccine 刺激之后，当时我应该受过那个 iris imaging data， 它是全身都在。而且肿瘤里面会增加差不多10倍的 CAR-T 在里面，因为有这么多的 CAR-T 在里面的话，就是 macrophage，似乎至少在我们本身研究来看的话，就是 macrophage 没有起到，因为它呢只有非常那种mild 表现的变化，相对于就是说肿瘤本身的那种 dendritic cell， 就是那种抗原提成细胞，类似于这种 migratory dendritic cell， 可以直接就是捕捉到抗原，然后同时 提升抗原的这种细胞的作用的话稍微少一点，然后我们没有看到就是说有明显的就是说效果，所以说我们就没有往那边继续去研究。当然了，因为我们做的可能只是有2个模型，一个是在文章里面我们主要是做的这个 CD2 glioblastoma model， 我们还有一个 melanoma 的 model， 就是黑色素瘤的模型。现在就是我们发现的只是在这2个实体瘤里面，如果换到其他实体瘤里面，可能那个 microphage 的效果可能就有点不一样。当然我们没有做那方面研究，所以我没法做更详细的就是评述在那方面。
好，谢谢您。对，这个是非常 interesting。对，我觉得它不光是 present，它自己也会被激活的，所以可能它
是的，是的。
M1、M2 可能都会有改变，但是确实你说的这是非常的就是 relevant 和一个something to follow up later， 非常感谢，谢谢，谢谢您的分享。
这个很好的讨论。那个我想，我们线上啊，因为我们研究所啊也有一些在从事 vaccine 这个研究的，我不知道大家有没有兴趣，有没有要问的问题？还有我们肿瘤医院那边，我看也有一些这个临床医生啊，这个研究人员。刚才我看那个赵月老师在。这样，我自己先问一个问题啊，就是说，
好的，
就是因为在这个 CAR-T 治疗这方面呢，在2013年就是给了大家这么一个欢欣鼓舞的一个消息，能够把这个血液瘤能够治愈，这个这么多年大家就一直在寻找实体瘤，让它这个提高效率的这个方法，就是说，作为您这个比较在这个领域里边这个耕耘多年的一个研究人员，对未来怎么看这个实体瘤的一个治愈？未来10年。
对，这是一个非常好的问题，也是我们我现在实验室的一个主攻的方向，就是对于血液瘤来说，包括尤其是 B 细胞方面，因为他们已经有了一个非常好的抗原，就是 CD19， 但 CD19 它也会丢失。就是血液瘤方面来说的话，你基本上你不用太担心，就是说 T 细胞浸润的问题，就是你怎么能够进入到实体瘤里面，因为本身它就已经是属于那种在同一个compartment 里面共存的状态。但是在实体瘤里面的话，就是说你很难找到像 CD19 这样的一个非常完美的抗原。那这样的话就给你一个很大的问题，就是你怎么来让这个 T 细胞来识别这个实体瘤。另外一个问题就是说，是这个 T 细胞是不是也会像这个在血液里面会有这种 tonic signal 或者是有这种 exhaustion？ 当然这几乎是肯定的。那这就给我们就是2方面一个主要就是说研究的方向，一个是如何让这个工程改造的这个 T 细胞来识别肿瘤，另外一方面是如何防止进入到肿瘤里面的 T 细胞有这种 exhaustion。那这样的话，就是像我实验室主要研究的是如何来让这个 T 细胞来识别这种肿瘤细胞，来多种多样的肿瘤细胞。就是说，但是现在就是说我现在发现就是说，如果是就是说能够找到这样一个方向的话，其实是能够真的能够把这个工程改造细胞能够有一个就是说很大的突破，能够就相当于让这个领域能够就是说重新找到一个方向来改造这个工程细胞能够让它来识别更多的肿瘤抗原但是又不会说去那个影响到健康的这种组织，因为现在就是可能是一个很大的方向，就是用合成生物学如何让这个 T 细胞来识别2到3个，而且只有同时识别2到3个这种抗原的时候，才会识别认为这个是一个肿瘤细胞。但这又带来另外一个问题，可能就会导致很多的复杂性的问题在里面，就是比如说，如果你丢失其中一个，那这个 T 细胞可能就不会再识别这个肿瘤细胞了。那这样可能会导致另外一个问题，你如何来改造你的 T 细胞，能够让它识别多肿瘤抗原，但同时又是的确这个是肿瘤细胞，而不是说是一个健康的细胞。所以说在如何让这工程改造细胞识别肿瘤方面是一个很大的问题。另外一方面就是像我刚才提到这个如何让这个 T 细胞能够防止它的 exhaustion。但是在这方面，其实很多的研究已经在这个血液瘤里面已经有很大研究了。其实这种信息你是可以完全转移到这个 抗实体瘤的就是工程改造细胞里面来的。所以基本上，像你说的，有未来10年的话，可能就是说如何让这个T细胞能够识别，就是说实体瘤能够更有效的识别实体瘤，而且又不会就是损伤健康组织，然后另外的话就是如何能够把我们从血液细胞里，从血液瘤里面学到的这些信息，如何来改造这种工程细胞，改造 CAR-T，让它能更有效的长期存活在实体瘤里面，来达到这么一个就是说彻底解决实体瘤的问题。可能我认为就是可能在未来10年，可能这是两个非常大的方向。
对，这个很好。我还有另一个问题，但是我先往后放一放再问。我那个看到这个非常荣幸，今天谭院士也在线。这个谭院士，您有没有这个要讲的、想讲的？谭院士。那这样，我再接着问这个问题，就是说，在这个肿瘤免疫治疗的这个过程中啊，它有一些表征，就是说你肿瘤是这个疗效是在还是不在，就是说你可以通过影像学来看，是不是还有一些其他的 marker 能够帮助你更快的能监测到你的这个治疗效果的，是吧？这些 marker 呢，目前常用的是哪些？就是说我们也想在这方面。在这个疗效监测方面，看看能不能做一些工作。
像这方面的话，其实我的研究不是很多，但是就根据我了解来的话，就是说，因为每个肿瘤可能，因为现在就是说，就是临床上他们检测一个肿瘤在不在的话，比如说他们会用 MRI 去检测，如果是脑瘤的话，他们会用 MR 去检测是不是在那个位置。至于那个，然后会有其他，你如果说血液，就是说直接从血清里面检测，或者是血液的那种信息的话，可能对于每个实体瘤它会有不一样的。但是对于如果是就是说因为我是原来接触过很多用这种工程材料来检测的，他们会有一种把很多的这种肿瘤里面非常有特异表征的一些酶的那种受体，或者是一些酶的一些一些可以有报道的这种我应该这么来解释，就相当于是一个检测的一个平台，就像那个纳米材料检测平台。你把这个纳米材料就是说输入到体内之后，如果是没有肿瘤了，那你就不会有它相应的就是说那种有那个信号。如果是那个继续有肿瘤的话，那就会有一个非常强的信号。这个可能就是就是说可能会用于更多实体的检测。但是普通的这种传统意义上检测的话，其实还是也是可行的。比如说你用那种MR 去检测这种脑瘤的话，这种其实是临床上经常来应用的这种方式。
挺好，我们也在考虑一些是不是非侵入的更频繁的这种，当然这要将来要再仔细的去讨论这一方面的是吧？除了影像学，有时候影像学你很难看出它来，它肿瘤这个体积的一个变化，它可能有时候是从中间开始坏死的凋亡，可能不能及时的进行一个判断，是吧？
对，
这个方面可能将来再考虑一下，跟您再沟通。另外呢，就是说我们线上啊有老师想问问题，是让我来帮助提问一下，就是说，他说佐剂在 CAR-T 活化中是否占主导地位？这是第一个问题。第二呢，就是佐剂对 APC 细胞活化显著，对 T 细胞不同亚型有没有直接活化的证据？第三呢，就是佐剂放大免疫会不会带来潜在风险？
你能回到第一个问题，重新再给我读一遍吗？认为
佐剂在 CAR-T 活化中是否占主导地位？刚才您也提到，
对，
佐剂是有一些会有一些这个带来一些效果啊。
是的，佐剂的话就是说它是一个，因为完全看这个佐剂你是怎么来就是说导入体内的。像我们是我们有不同的佐剂，我们都尝试过。其实因为这个佐剂我们最常用的是让它直接进入到淋巴结里面，那就是它不会这种自由的释放到体内，所以说不会产生很多其他的这种负面效果因为只要进到淋巴结里面的话，这就是 T 细胞还有正常所有的免疫细胞的存在处，这些佐剂会被一种就是淋巴结里面正常的这种抗原器正细胞所捕获，然后激活它们。 而佐剂的最主要的效果就是激活这种抗原 T 细胞，然后让它们去提成这些共刺激分子，然后释放这种细胞因子，然后来影响 T 细胞的分化。当然，如果不同的佐剂，它有可能会导致不同细胞因子的释放，这样的话就有可能会导致 T 细胞在激活方面或者分化方面有不一样的这种分化趋势。但是因为我们是用于 CAR-T 方面，就是 CAR-T 本身已经是在体外被激活了。然后你再放到体内的话，它的就是说整个表型可能并不会像在体内就是说完全那种原始 T 细胞被激活的那种状态一样。我不知道这个能不能回答第一个问题。对，
对，其实是这老师是我们研究所的一个老师，不知道他是不是在线。如果在线的话，张老师你在线吗？他可能是通过其他平台在听，
是吧？
因为我们那个张老师你要在的话，你再接着自己问。他在线可能是在其他平台，并没有在我们他在其他平台并没有在我们这个腾讯会议当中。好，我帮他再问第二个，他就是说佐剂对 APC 细胞活化显著，对 T 细胞不同亚型有没有直接活化的证据？
这个的话就是说它会有影响，但是我们没有做更多深入的报道，因为也就是没有，就是对我们来说没有做更多的这种深入的研究因为我们知道就是我们所有的就是说表型的这种研究的话，全都是在体内。就是说其实如果看 T 细胞被激活的话，包括它内源的一些这种效应的分子，像 granzyme， 像这种granzyme， 还有介素的释放，还有TNF alpha、interferon gamma 它们所有的释放，还有一些所有的一些代谢方面的研究的话，其实我们都是有明显的就是显著的增强。比如说像我其中提到过一个做了 RNA 测序，然后发现他们很多的这种一种 gene expression signature 会倾向于就是说代谢方面的一些重编程，可以导致了一些 T 细胞可能在很多方面代谢方面有绝对的增强。然后最近其实是在差不多1年前有一篇 Science 报道说，就是因为代谢方面的增强会直接影响某一类的细胞因子的释放，就包括interferon gamma。所以就是这个很多方面其实是我们并不能明确说这个佐剂本身会对 T 细胞有很大的影响，但是这个佐剂对 APC 的影响之后，然后这个 APC 跟 T 细胞这个 CAR-T 正向接触，就是它有一个我们称之为 crosstalk， 这样的话就是说能够让这个 APC 接触到就 CAR-T 能够就是接触到这个 APC 给的所有的刺激，这样的话其实才是一个就是说最强就是佐剂最重要的作用所在。
对，我想他的第三个问题就是佐剂是否会带来这个潜在风险，也已经回答了。对，那目前我们线上的许教授，
宋老师，我可以提个问题吗？
你好，刘老师，刘老师也是我们研究所的一个在做这个肿瘤疫苗的研究的一个这个研究员，我们的研究员。
马老师好，非常感谢今天上精彩的报告。不好意思，因为我刚阳，这个形象不太好，我就不开那个摄像头了。今天非常受益这个报告，就是您讲到的，就是您这种增强这个 CAR-T 的这个疗效，看到非常好的一个手段。就是现在我们也看到有很多研究也在开始做这种原位的 CAR-T 治疗。如果您这种手段跟这种原位的 CAR-T 治疗最后进行联合，是不是也会有一样的这样一个增强的效应呢？或者还是说在某一些地方会有一些不同的需要进一步调控的？想您对这方面跟我们您的看法。
你说的原位的 CAR-T 治疗是在体内，就是说生产这个 CAR-T 吗？
就是现在比如我们做就是比如说他们直接把这个 mRNA 的这个纳米制剂打到体内，让它去靶向 T 细胞之后，让它生成表面这种CAR。因为这样的话有点跟您那个佐剂就是相当于都是通过外来的这种有点像药物制剂的方式给打进去。
对，其实这方面就是说我知道很多人在做这方面。如果是说是 mRNA 方面的话，其实我们这个佐剂是 不是好的效果，反而会造成不好的效果。因为 mRNA 它是短暂表达的，如果就是说这个 T 细胞真的能够表达这个 CAR， 大量表达这个 CAR， 然后接受我们抗原刺激的话，其实一旦 T 细胞一扩增，这个 CAR 就基本上就会丢失了。因为它就是说在扩增的 T 细胞，就是 T 细胞是一个非常强的扩增能力，一旦接受这个疫苗刺激的话，那就基本上是说，一旦 T 细胞扩增完了，它这个 mRNA 基本上也就消耗殆尽了，就不可能再有更好的这种，至少在肿瘤方面没有更好的这种效果。当然，如果是你可以用病毒能够把一个就是说 CAR 这个表达基因给就是完全整合到 T 细胞的基因组里面的话，那这个是我觉得是跟我们这个疫苗实际是完美的搭配。
好的，非常感谢啊。
很好很好。那个我看已经接近11点半了哦，我们再问最后一个问题吧。我刚才看这个许教授在开视频，这个
好啊，行啊，刚才大家提了很多问题，我觉得都非常好。那我还有一个问题啊，想问一下马教授，比如说主要是这个 deliver 这个系统，不管是就是因为我们知道在这个室内瘤啊肿瘤环境当中，它有很多的免疫调节细胞，包括我们叫 应该我们讲 TOT 啊、TOB 啊、TOPC 啊、T macrophage 等等这些都有，对吧？那么就是说一个问题呢，就是说，你这个疫苗呢，它跟 mRNA 疫苗之间的，就是说在比如说临床应用当中，它的安全性和它降解问题，比如说一些到体内的降解的程度，到这个周围淋巴结，然后呢，再到肿瘤浸润地方的那个 DC，这个之间你有没有做过什么研究或者比较呢？
其实这是一个非常好的问题，因为就是说我们当时也想过这个问题，但是因为我们就是说之前没有做过 mRNA 研究，就是至少是前几年，就是最近的话，因为我自己的实验室刚刚建立起来的话，这是其实我问的第一个问题，就是说因为 UPenn 它也是就是非常强的，就是 mRNA 就是说研究所在那边，然后我们就是有很多合作，就是其中一个合作方向就是通过这个 mRNA 疫苗来。就是说表达，比如说这个 EGFR3，就仅仅是这个抗原在那个就是在通过纳米材料就是说打入体内之后，让它在这个抗原 T 成细胞里面表达，然后来比较我们这个就是安非法外这种双亲性的这种疫苗。其实现在来就是说初步结果来看的话，其实因为两种疫苗它这个在体内的就是说传播方式或者是那种你说的 deliver， 就是说它会到体内什么地方是完全不一样的。像我们这个两剂疫苗的话，如果是你在皮下注射，它基本上只会在淋巴系统里面，就是说传播，不会跑到血液里面。但是你如果打入这个 mRNA 这个 lipid nanoparticle 的话，它会到 liver 里面，会到 spleen 里面，会停留在 muscle 里面，就是说它也会有一部分会进到这个淋巴结里面。但这是其中一个非常大的不同，然后这样的话就可能会导致 CAR-T 会直接入侵这些表达这个 就是说被这个 mRNA 这个 lipid nanoparticle 转染的细胞，这样可能会有一个二次伤害。另外的话就是说，两者比较下来，对我们就是说出现的结果研究结果来看的话，其实是我们这个两剂性的疫苗，其实有绝大部分两剂性的疫苗能够被插到这个抗原 T 成细胞表面，而且就是另外一种，就是从另外一个角度来说，其实也是一样的，就是说绝大部分的这个抗原 T 成细胞也都会能够提成我们这个就是说双亲性分子，但是对于 mRNA 疫苗的话，其实只有很少一部分的这种抗原 T 成细胞能够就是表达这个。但是另外一点不同就是说，因为你是 mRNA 去表达，所以你在抗原 T 成细胞表面所停留的时间要长一些。所以这有可能就是弥补了它，就说即便有很少的抗原 T 成细胞来表达这个分子，可能也会有相对长时间的刺激。但是整体来说，就是说后续研究的话，其实我们还没有完全拿到我们要的结果，所以现在我不便于就是过多的评论。但这是就是两个疫苗平台，就是两个疫苗就是说方式，就是整个在体内的转染或者是那种传播方式，还有细胞表面的这种提升方式的不一样之处。对。
好11点半那边这个美东那边也很晚了，那我们今天就到这儿。这个我们韩老师，你有没有想讲的？给大家总结一下。韩老师，那这样， 那我们就这样保持沟通，这个马教授。10
可以
月份你来回中国的时候呢，一定要联系我们，如果方便，可以线下来杭州呢，到我们研究所来进行交流。第一，我们是肿瘤医院，这个肿瘤医院呢，我们是一体化发展和这个本来我们叫这个中科院基础医学与肿瘤研究所啊，这个有很多的老师团队呢在做相应肿瘤免疫治疗，还有基因免疫治疗啊 mRNA， 这个免疫治疗方面的这个工作，大家我想有很多共同的兴趣啊，所以我们保持沟通。10月份呢，希望能这个线下交流来我们研究所，好吧？
可以，好的。好，
那我们今天就到这儿，就交流到这儿。这个感谢线上的各位老师同学，感谢这个张教授，这个许教授，张放博士，感谢你们参与。好，谢谢。
好，拜拜，
保持沟通，拜拜，再见。好。